All right, my mic's ready to go? Good volume? Okay. Hi, everybody. It's a pleasure to be with you here today and talk a bit about some of the work that my lab is doing in the area of RNA delivery. We are much more on the fundamental side of things as opposed to the talk that we've just seen from BioNTech founders. And really what I wanted to talk about today is the unsung hero, in my opinion, of the COVID-19 vaccines and what will continue to be super relevant as more and more mRNA therapies roll out. And that's these fat balls that we call lipid nanoparticles. So today I want to tell you a little bit more about what these nanoparticles are, what the ingredients are within them, which are the ingredients within our vaccines, and a little bit about how my lab thinks about the chemistry and how to improve the efficacy of these particles for future use. Let's see. So those of you who were here from the last session, this was covered to some extent, but I'll repeat here in case anybody has come in since then. So messenger RNA is part of the central dogma of molecular biology, which tells us that our DNA, or a permanent blueprint of genetic information, is then transcribed into messenger RNA, which is essentially this middleman that hangs out in the cell and then is translated into protein. So proteins are the doers in our body. They are responsible for nearly all functions that we have. And when we develop different types of disease, it almost always includes some kind of malfunction with protein expression or protein function itself. So instead of trying to directly control protein, the amount of protein or the type of protein that we have when we develop therapies, one nicer way of doing it, in my opinion, is to go after the genetic information at the messenger RNA level. And that's because if you want to deliver, let's say, 100 proteins, you have to get 100 proteins into the body. Whereas if you deliver a single molecule of mRNA, that can then go on to make 100 proteins. And so the effect can be quite amplified with this approach. Another nice thing is that when you express mRNA, it's a very temporary event, and that can be really nice for certain types of therapies where you want the effect to be acute, and then you want to move on. So what my lab does is we think about manipulating protein expression by manipulating mRNA, and you can do it in a couple of different ways. One of them is to use short interfering RNA. This RNA molecule has been around for longer, and it was discovered in about the year 2000. And we can use this particular small RNA to silence or knock down protein expression. It could be proteins that are mutated and non-functional. It also could be proteins that are overexpressed in certain types of diseases, and we want to decrease expression. More of the highlight of my talk today and what we just heard about is messenger RNA delivery. A whole lot of science has been done to design messenger RNAs to get them to the point where we could even think about delivering them and developing a therapeutic. But in this case, we can augment mRNA expression that we already have to make more of a particular protein, or in the case of vaccines and some other applications, we can introduce messenger RNA that encodes a protein that our bodies have never seen before, and to make that to induce specific effects. So although a lot of focus has been on the vaccines lately, there is such tremendous therapeutic potential in messenger RNA, which Ugur had also mentioned. So in addition to the vaccines, which we think about for a while, there's also been a lot of effort into protein replacement therapy. So in this case, you're trying to make more of a protein that we need. Some examples of potential treatments would include cystic fibrosis, muscular dystrophy, where we have mutated proteins and really we need more of the correct version. There are cancer immunotherapy applications. cellular reprogramming applications, so you can direct the differentiation of certain types of progenitor cells with the presence of proteins called transcription factors. And so if you can express those factors, you can direct differentiation. And now all of these applications have to do with upregulating a particular protein of interest, but we can also use messenger RNA delivery to downregulate protein expression through the use of gene editing. And this would happen by delivering messenger RNA encoding one of the nucleases, for example, Cas9. So if you'd like to hear any more about the applications for this or delivery methods, in addition to what I speak about today, you can refer to the review I have here. Now, despite this tremendous therapeutic potential, we have a problem when it comes to messenger RNA. It is very large. It is uncharged, and so if you try to get it across our cell membranes, they are also charged and they will not take it up. And it also has really lousy biodistribution properties in vivo. So if you were to inject mRNA by itself into the bloodstream, it would very rapidly be cleared by our immune systems. And because it's so unstable, it will also degrade. And so what messenger RNA needs is our help. It needs to be packaged up in a material that's going to protect it, but also to help escort it across numerous challenging barriers inside of the body. So here we have a schematic of what some of these barriers will look like. So here we have the particle itself. In the case of an IV injection, we would inject directly into the bloodstream. With a vaccine, we're going to be going into the intramuscular space. These particles then have to move through the bloodstream, avoid being uptaken by the macrophages and other immune cells that are in the immune system. Step B there is they need to extravasate to move out of the blood vessels and diffuse through the extracellular matrix to approach that target cell of interest. And then step D is to get into the cell itself. Okay, so the lipids, they kind of look like the lipids of our cell membranes, and so that's part of why our cells are willing to take them inside. But when they take them inside, they keep them in this walled-off compartment called an endosome, and that endosome is there to help the cell decide, is this something I want to take in fully into my cytoplasm, or is this some kind of garbage or junk that I need to degrade and then get rid of? Well, normally your cell, when it interrogates what's inside that endosome, it sees a lipid nanoparticle, it says, this is not for me. So a lot of the design of these lipid nanoparticles involves the incorporation of chemistry that can basically fuse with that endosomal membrane, kind of against the will of the cell, and to help that particle get out into the cytoplasm of our cells, where finally it'll unpackage and interface with translational machinery to make that protein of interest. So years ago, my lab asked the question, can we develop potent and degradable nanoparticles that are capable of overcoming all of these barriers? And why potent? Well, that's fairly obvious. But degradable because many mRNA delivery applications, therapeutic applications, are going to involve repeat administration. And so while degradability isn't necessarily a huge issue for one-time injections, it is a big issue when you have things accumulating over the body, in the body over time, which I'm sure you can appreciate. So we work with materials that we call lipidoids. Lipidoids simply means a lipid-like material, and we designed them to mimic legacy lipids that have been used to deliver different types of nucleic acids for a long period of time. So here is a cationic lipid that we call DOTMA. This has been used for decades now to deliver originally DNA and now other types of nucleic acids. And this material can form, the legacy form of these nanoparticles were liposomes that look very similar to our cell membranes. They have an aqueous core where you can package different aqueous types of, hydrophilic types of drugs, including nucleic acids. So what we want to do instead is to try to make materials that look like this. However, nobody really understands very well how the chemistry affects the functionality of these particles. So we wanted to make large libraries of these lipid-like materials and really screen them to understand, to try to then get structure-function relationships out of these data so that we could better understand what kind of chemistry do we need in a nanoparticle to promote functionality. So here's a little chemistry for anybody who likes chemistry. We do what's called a combinatorial reaction, where we take a group of what are called alkylamines. So in orange, you can see there's a group there called a primary amine, and that is going to react readily with the molecule in pink, which has an acrylate at its end. And so those materials, if we mix them together in a vial, we don't need any solvent. It's very easy to make. So out the other side will come these lipidoid materials. And the specific chemistry that we've used helps with its degradability. So here you can see in blue, we form groups called esters, and these groups will readily degrade in the presence of water. So this will break apart into smaller components that our body then knows how to handle in terms of clearance. So how do these materials then help to form nanoparticles that encapsulate our RNA of interest? So normally these lipids are going to be neutrally charged at a neutral pH. So when we inject them into our bloodstream or our muscle space, they will be uncharged. And that's a good thing because that does not promote toxicity. Positive charges do. So it's nice to avoid those positive charges in the bloodstream. However, once these nanoparticles get inside that endosome, inside the cell that I told you about, the cell actually starts dropping the pH within that endosome on the way to degrading the material. And so these materials, these lipidoids, as we drop the pH, they begin to take on this positive charge, which is really helpful to help them get out of that endosome into the cytoplasm, but it's also really helpful to pull in our negatively charged messenger RNA because these really nice electrostatic interactions. So when we make the nanoparticles, we basically have an RNA stream that's dissolved in saline or buffer, and we mix that with all of the lipid ingredients in our nanoparticle dissolved in ethanol, rapidly mix them, the nanoparticles form, we dilute them in buffer to help stabilize them, and out the other side of, in this case, a microfluidic device, here's what these particles look like, approximately. Nobody really fully understands the internal structure of these materials. However, they contain both hydrophilic regions and hydrophobic regions. And there are 5 main ingredients, 4 in addition to the messenger RNA itself. And they each have very special roles inside of the particle. So the one that we've been interested in developing is number 1, the ionizable lipid. This is the proprietary lipid in most cases. That's because this is one of the only ingredients in the nanoparticle that has to be made in the lab. Okay, this is not a naturally occurring lipid, and that's why it's important to design it in a way that's degradable. So Moderna has developed its own ionizable lipids. BioNTech had their own. Most academic labs have their own, and so that's that. Cholesterol, we have a ton of cholesterol in our own cell membranes. We would be a bunch of goo without it. They help to stabilize lipid bilayers, and they do the same thing within our nanoparticle. They provide stability. There's another molecule that we call a helper lipid. This is a naturally occurring phospholipid that we can also find in our cell membranes. This is there to help with the organization of the lipids inside the nanoparticle. And depending on the size of mRNA, for example, that you're working with and the type of ionizable lipid, you might choose between a couple of different types of naturally occurring phospholipids to help all these lipids pack in exactly the right way inside of the particle. And then finally, there's a PEG or polyethylene glycol lipid. This you can see, it's the gray squiggly molecule on the outside of the nanoparticle. PEG itself is hydrophilic, and so it doesn't want to get inside of that generally hydrophobic molecule. It decorates the outside and it provides stability. We actually consume PEG. Some of us consume PEG. It is the only ingredient really in Miralax, and it's also found in many common household and cosmetic products. Some of you have, probably all of you have heard in the news about some of the allergic reactions that have developed in response to the vaccines. Although the science isn't entirely clear yet, there is mounting evidence that the polyethylene glycol is responsible for some of these reactions due to prior exposure to the material. Now, why is it happening in only a small portion of the population? Everybody's immune system is different, right? So just the way that some people develop allergies to latex, other people are going to have allergies to PEG. So these particles ultimately, here's an electron microscopy image of them, they tend to be about 80 to 100 nanometers in diameter. And so over the years, my lab has screened thousands of these ionizable lipid materials for RNA delivery. Here's an example of a library that we have developed. So here we had 5 different tail groups, those acrylate tail groups, and 280 amines. And so we react each of those with one another, and we wind up with a 1,400 member lipid library, which we can then go on and test in cells and in mice. So originally, we've developed many of these materials for siRNA applications. That's because it was around as a therapeutic for about a decade before mRNA became viable as a therapeutic. And looking at these different lipidoids that we developed, we've been able to deliver to many different cell types and organs throughout the body. So we've had a lot of success, really, with these materials is what I want to convey. So then when messenger RNA became viable, we wanted to ask the question, can we use some of the materials that we had previously developed for siRNA, can we use it for messenger RNA delivery? In many ways, we thought this might work because siRNA and mRNA are made out of the same building blocks. However, it's much more complicated than that, and messenger RNA is more difficult to deliver because these two molecules are molecularly distinct. So here's siRNA. It is a double-stranded RNA where each strand is 21 bases long. So it essentially behaves like a short, stiff rod. Okay. Messenger RNA, on the other hand, is single-stranded, and it's huge compared to siRNA. It tends to be anywhere from, say, 1,000 to 4,000 bases in length is a typical size. And you can see here all the curls. It takes on both secondary and tertiary structure that makes it much more difficult to package it up inside of a nanoparticle. So basically what we've seen is that only a very small subset of the materials that worked well for siRNA are effective with this more difficult molecule, messenger RNA. So when we first got started in the messenger RNA space, we decided to look at a family, a very small family of 11 lipidoids. And in this case, we reacted one of our very, very best amines from our siRNA work, this amine that we just called 306. And then we reacted it with 11 different tails to try to understand how the tail chemistry affected the ultimate functionality of these particles delivering messenger RNA. So we assessed our in vivo efficacy using an imaging technique called IVIS imaging. We work often with reporter messenger RNAs. When they're delivered properly, we're able to see the expression of a particular protein. So for this work, we delivered messenger RNA encoding firefly luciferase, just the way the fireflies will light up at night. If we've successfully delivered this protein, we too will see luciferase signal. So we use this big IVIS imaging box to look for that signal. So here we're looking at this protein expression across a number of different organs, and this is naked mRNA. It means that it's not within any kind of delivery vehicle, and you can see we don't really have any signal. So then once we incorporate this messenger RNA into our lipid nanoparticles, you can see that they work. There's a spectrum through which they actually work. And there's this one material. You can see the bright red liver. We call it 306OI10, but the name is kind of meaningless in this context. This is so much better than the rest. And the one below it is still quite a good material, but it just pales in comparison to the one that we found up above. So this is simply quantification of the results, taking into account all the animals we studied. And you can see again that this top material is, there's a broken axis. It's essentially an order of magnitude better than the rest. And what's really awesome when you look at these materials is it just goes to show you how important the chemistry is. So look at the one that's not so good. It's a 10 kind of good. It's a 10 carbon tail and it's linear, okay? It's a straight chain. Then the one that's an order of magnitude better, it is the same molecular weight, it is the same chemical formula. The only thing we've done is to take the terminal carbon and to move it one position in so that there's a small branch. One carbon moved one position. It is one ingredient out of 5 ingredients in a lipid nanoparticle, and it works an order of magnitude better. Pretty cool. So this is our beautiful material that's come out of all of our screens. This is the one that's worked the best, and it's really good at doing things. So here we are comparing it to one of the industry standards, MC3. You can see that it works better than that particular material, which has been part of FDA-approved formulations. We also compared by histology and other types of toxicity. It does compare favorably. I'm not going to go into what these images mean. Another thing that unfortunately happens for a lot of delivery vehicles that we develop is that they work once in an individual and they don't work ever again. Well, that's really problematic. So we did testing where we looked at the signal of the luminescence we were expressing over a period of several days. You can see that the first dose and the second dose, which occurred 1 month after the first dose, they resulted in nearly identical protein expression, kinetics, and so that was also reassuring. And we also showed that this material is potent enough to co-deliver multiple messenger RNAs. Here they're simply reporters, so luciferase, mCherry, and then erythropoietin, which is secreted, and so we measure that with an ELISA. So same animals for all of these. So it's really cool that you can do multiple at once. So in summary of my work, and I'll talk just a little bit about how this applies to the vaccines, what my lab has found is an unusually potent material for messenger RNA delivery. We've seen that very small changes in LNP chemistry dramatically influences delivery, and I think it just is more evidence of the importance of some of the fundamental research that's going on mostly in academic labs to try to learn more about how do we make particles that are then going to be used by companies and are capable of translation. And we've also seen that our lead material is good for a number of things, multiple mRNA delivery, and also we can gene edit as well. So how about the use of lipid nanoparticles for these COVID-19 vaccines? So most lipid nanoparticles that people use across the world are incredibly similar. And so here are the same materials that I already told you about. They are present in the Moderna, BioNTech, and if there are other lipid nanoparticle vaccines on the horizon, they'll look similar. So the only difference between their two lipid nanoparticles are their ionizable lipids. So here is what BioNTech Pfizer's looks like. We were delighted when we saw this structure because notice it has branched tails and it has ester groups. So that's pretty confirming of the work we've done. And then Moderna, look at theirs. It looks very, very similar. So it's reassuring when independent groups of scientists converge on similar solutions, right? It just goes to show you that this is real. So for those of you who are not familiar with how this then ultimately gives us immunity to infection, we take our nanoparticle. It's going to enter the cells if we've designed our nanoparticle well, and it's going to release the messenger RNA encoding the spike protein into the cytoplasm. So the translational machinery will then generate the spike protein as well as tiny bits of broken down spike protein. This is even a simplified version of immunology, which is just extraordinarily complex. But the cell then says, hey, immune system, look at this. It shows the spike protein on the surface of its cell, and the immune cells come on over to see what's going on. So B cells, which have very specific binding receptors, can eventually there will be one that matches up with the spike protein. Those B cells will then start putting out antibodies and train memory B cells so that in the event virus enters our system, we'll have this virus, the antibodies will coat the virus and neutralize it and then clear it from the bloodstream. But then we have a backup mechanism. These B cells are then also going to train T cells within our body. All these cells cooperate with one another. They're all talking to each other. And ultimately, these T cells, called cytotoxic T cells, get the message that if they ever see an infected cell, that they are going to kill it. And so should the virus not be neutralized and actually infect one of our cells, the infected cell will present some evidence of the infection on its surface. The cytotoxic T cells will kill it. They will recognize it, and then they will kill that infected cell. So our immune systems are pretty remarkable. We have this backup system so that first it tries to prevent infection in the first place, and then if infection does begin to occur, it's going to kill the cells so that they don't spread it further. So that's why, the vaccines don't always completely prevent infection, but because of this backup mechanism, they have a great way of, when that infection starts to happen, to kill a lot of the infected cells, which results in many of the mild cases as opposed to the more severe hospitalized cases. So with that, I'd like to thank you for the invitation and for your time, as well as the members of my lab who have carried this out. You can follow me on Twitter, and just last month I gave a TED Talk at the annual TED conference talking more about some of these vaccines in even more of an accessible way if you'd like to look it up and share it with anybody that you know. So thank you.